on track. All right. So today, the basic topic will be the data processing part. So before we get into that, I want to overview a little bit what we have discussed last time. And so the first one is, what is next-generation sequencing? So there's a keywords about next-generation sequencing. The first one is sequencing, right? It sequences both the DNA and RNA with modified protocols. What I mean by modified protocol is this equipment really doesn't sequence DNA or RNA. You have to make it back to the DNA so that you sequence it. And the second keyword is short reads. So it can be 35 base pair. Actually, for the earlier platform, it's a 25 base pair, and to 100 base pair at the current stage. And some of other platforms, it can go to 400 base pair, which is the 454 technology. And so you can see that, you imagine this is the reads, and you do not sequence from the very beginning to the end. You only sequence the very beginning part of it, or both two ends. And that is called either mate pair or pair ends. And the third keyword is ultra high throughput. So we got 1 to 1.5 billion reads in one sequencing run, and that's a lot of information to go through. And we also talk about the leading platforms, and the first one is the Illumina platform. And this is really the leading platform. It takes about 60 to 70% of market share. And the amplification of Illumina platform is the bridge amplification, and the sequencing is the sequencing by synthesis. And the second most important one is the solid platform, which is also purchased by the Life Technology. And comparing to the Illumina, for the amplification part, it uses immersion PCR rather than bridge amplification, and it's sequencing by ligation, not really synthesis. And this is the color space, and which is a very unique feature. I hope you already went through the lecture notes, and those will be, hopefully it's clear for that. And the first platform, actually, for the next generation sequencing, I would say the commercial platform is the 454 platform. And we have this, they also call it peri-sequencing. This is because of the biochemistry, the way they sequence. And the major feature of this is the longer rates. So it's 400 base pair rather than only 50 to 100 base pair. So remember, I have one slide last time to show you the cars, the cartoon. So you break the puzzles into bigger pieces or smaller pieces. And one of their selling point is if you're sequencing longer rates, it's really easy to recover what you really sequenced to begin with. And this, as I mentioned, this platform came in commercial from 2006, and I believe that Illumina is from 2007, and Solid is from 2008. So they were 1 year apart from each other. Another major player is the Pacific Biosciences, and this is a single molecule sequencer. So they skipped some of the amplification procedure, which make it have a lot of good features that people can work with. And it's also the nano hull. There's one lecture notes, if you remember, there's a hull container there, and the polymerase is sitting inside it, so you can see the DNA molecule going through it. And while the polymerase is going through it, it starts to excite the lights so that we can read it. And but the real feature is it's a single molecule, and it's nano hull, and it's very long rates. This is really unique rates, unique feature, and it can go to 3 to 5 kilobase for each rate. So it's very, very nice. And the last one, but probably you will see the market share goes up for diagnosis market, which is L02. And this is the personal genome analyzer. So it's not, we're hoping that in the future that every lab can have this kind of sequencer sitting on their bench, and if you run some small samples, it's really doable. And L02 is one of those machines to target those kind of market. And the machine itself is very cheap. It's only $50,000. So the trick is they skip those fancy, more expensive camera part. So rather than use a semiconductor-based technology, iron system semiconductor is make the machine much cheaper. And so far it's lower throughput, but it's very, very fast. So you can get a reasonable amount of sequences in an hour or two. And so far, the error rate for the error current is a little bit higher comparing to the Illumina and Solid. Last time, one student asked, I believe it was Kelly, asked what's the error rate of that. And I heard the rumor it's about 10%, but when I go back and look at their manuals, actually it's, the accuracy is 99%. So it's not too bad. It's pretty good, reasonable. But at least that's based on their commercial flyers. I don't know how real that is, and I think it's remain to be tested. And this one is a summary table, and you cannot see that very clear from back, but this is what I just mentioned, the major features of major platforms. So these are the things, if you want to look for a job in this area, those are something that you need to be familiar with. Okay, and also from last time, so what we can do is this technology. The first one is to sequence the DNA. We can do the de novo sequencing or reference-based resequencing. For us, this is a major application, all right? So your goal of here is to identify the genetic variants in the samples you are sequencing. And it can also sequence the RNA, which is called transcriptome-wide sequencing. And we will have several lectures about it. And this is really trying to get into the market share of microarray technologies. We know that microarray is used to measure the gene expression, and RNA sequencing can do the same thing and can do even better. And the third one is to study the protein DNA or protein RNA interactions, and which is the ChIP-seq or CLIP-seq technologies. And the last one is we can use all this to study epigenetics, which include the DNA methylation, histone nucleosome positioning, or chromosome looking. And I also spent almost half of lecture to review that the applications that were enabled by this technology in recent years. And the first one is the 1000 Genome Project. So the basic goal of 1000 Genome Project is after we have the reference genome, we want to characterize the variants in our human in different populations. And the Cancer Genome Atlas and International Cancer Genome Consortium, both of them are trying to understand cancer from multiple domain information, including the DNA, RNA, methylation, histone modification, and most of the things that you can think of. And these are two larger consortium. And also ENCODE project and mode ENCODE projects, those are the basic goal is to annotate our genome. Where did the noise come from? And I also introduced two concepts, which one is the mate pairs and pair ends, and they look the same. You got DNA fragments, you sequence both two ends, but they have very subtle but important differences that you can go over that. And multiplexing and barcoding is an important thing that we can keep in mind because of those that we can adequately or effectively use the resource that we can get from next generation sequencing, because so much data, we don't need that for one individual sample, so we can break one run into multiple samples through barcoding procedures. And the challenges is the data analysis, which is the bioinformatics part, and also briefly touch base a little bit on the cloud computing part. So just spend 10 minutes also just to run through what we have last time, and if you have any questions, send me email, and I will try to address those. Okay? And today, the lecture is not going to be as exciting as last time. So we need to go to very detail about the data processing part. I will first talk about the data analysis workflow, very brief introduction, so break the analysis into different pieces. And also, the first thing I want to talk about is sequence quality evaluation. Okay, this is it's not really very shiny thing that is how great this technology is. We want to break down, we want to identify their weakness so that we can use them more effectively. So there's a lot of sequence quality evaluation, Phred scores, and NGS error rates. And one of the major tasks today is to go through the alignment theories. How many of you have taken any type of bioinformatics class? All right, most of you have. So I'm sure that alignment is a major thing that probably is one of the first things you talk about in the bioinformatics class. And everybody is familiar with the Smith-Wortman although I'm still going to talk about it because we have students from School of Medicine. And also, one of the major things that I want to go through is the short race aligners. And I'm not going to go into detail of any one of it. Rather, I want to summarize the information, what type of alignment algorithm is available, what type of strategies they are taking. And one of the things I want to talk about today is Burrows-Wheeler transformation. And this is becomes very important in alignment algorithm right now. And I want to make sure that you know what is that. And we will struggle together. And to be honest with you, before I organized this class, I heard about it. I don't know what that is. So it's pretty fun to figure that out. And also, actually, it's one important thing is to compare different aligners. So there's so many different alignment algorithms, which one to pick, what are the pros and cons for each individual one, and those are the things I'm interested in talking about as well. Another major thing that we probably will spend 45 minutes also to talk about is the data formats. So this becomes very detailed, and it's not really too much scientific driven, but for the students who are interested in analyze the data, and you have to know what different type of data are and what the data formats are, what they means at least. And the last part will be the data visualization, and I will briefly go over genome browser and IGV. And this is the part that you can pretty much relax because most of you are already familiar with this. But these are really powerful tools to help us to visualize the next generation sequencing data that we have generated. Okay, so data analysis processing workflow. Okay, so some of the students already seen this class. This is from one of our very, very early experiment. And I don't remember which year it was. So this is the data was copied, but I think it was generated in 2008. So by that time, so you can see that this is the file size, and this one is, one file is about 4.3 gigabits. So one text file, all right? And I want to see how many rows in this file. For this particular one, it's 38 million rows in this one file. And what happens is this is what the file will look like. So we got 38 million such rows, okay? And you got a lot of information regarding the sequencing. Each row corresponding to one read you get out from the sequencer. And also there's a lot of characters which doesn't make any sense to any human being. And those are represent the qualities. So these are the things that we will talk about today as well. And the data volume is really humongous. It's really huge. So there's several data stages. The first one is the image processing part. So as we know that most of the sequencers so far is still based on the image, Illumina, Solid, and most of them. Other than Aaron Torrent, I think they are all based on images. And those are huge, huge, huge images. It's very, very refined, very high quality, high resolution images and multiple layers of it. And after that, we need to derive the base call from those images, meaning that when this location for the first nucleotide is A, C, G, or T, we want to identify those. And those are called the base call part. And after that, we still need to do the genome alignment. Okay, for the image part, for the high-seq technology, and this is not the latest biochemistry of high-seq 2000, actually. The latest one is 2000, I think it's a 350G run. And the image data is 32 terabytes, just for one run, okay? And in our CCBB right now, our storage capacity probably is, we have 2 backup servers. Each one is 54 terabytes. And I thought it was pretty big. And apparently that one run of a sequencer will take most of it if we really store this image data. And the intensity signal is about 2 terabytes. Base call quality data is about 250 gigabits. And alignment output can be 1.2 terabytes if we got all the intermediate files removed. So you can see this is the size of the data we're talking about. Actually, if you talk about this level, alignment, output, or even the base call level, we can store those. So those are fine. But for the earlier part, it's a big challenge. And this is a very early run. So for the Illumina GA2 50G run, so image size is 5.6 terabytes, and the alignment output eventually is 300 gigabits. So those are still big numbers, but comparing to these 32 terabytes, and it's a lot better. Okay, and so as I mentioned, the first level of data is actually the image data, the raw image data. They are very, very large. Okay, so 32 terabytes for one run. But the issue is here, should we really keep it? Okay, we know that a lot of institutions, they do not keep it, and we do not keep it as well. So once we generate the data, we calculate the base call, we throw the raw image away. It's just too expensive to store those data. It probably is as expensive as you regenerate the data. Okay, and there's problems with that. The major problem is when the sequencer company, when they updated their software to derive the base call from the raw image, and your later calls and before and after the switch, and they are not comparable anymore. And that happened once for the Illumina from 1.3 to 1.4 version change. And that happened once, and a lot of institutions, they were really panicked at that moment. But what you can do with it, and there's a data is just too expensive to store. It's more expensive than you regenerate. So a lot of institutions actually, they keep the samples, and if they need to get the data back again, they regenerate it. And process the data for the BAM file, and we will talk about that. It's about 1 gigabase for every 20 million sequence rates, and it's much acceptable. So it's those are the level of data we will really save. And for the data transfer, if you think of data storage is such a big issue, data transfer is impossible, right? If we do some experiment, we send the big samples to Beijing Genome Institute, which is across the world, and you want to transfer that data back, and through internet, that's a lot of bandwidth to really transfer those data back. HTTP and FTP protocols won't work anymore. And some advanced computer technology companies, and they develop the commercial software for protocol become important. And there's a protocol called next generation file transport, and this is the speed that they will get when you transfer data from long distance. And so far, the best technology we are using is a hard drive. Put the data into a hard drive and FedEx and send to us. So, and it only takes overnight. It's very secure and very fast. We had that a couple of weeks ago. We want to transfer some data. It was MCF7 DNA sequencing data. We want to transfer that from Ohio State back to here, and it takes about 3 weeks of transferring. And we gave up, so we still eventually get the data from hard drive. Okay, so a lot of things going on, a lot of information, a lot of number crunching, and but if we break the analysis into smaller pieces, actually these are the steps. It includes the primary analysis, the secondary analysis, and the tertiary analysis. I know these names doesn't make any sense. I didn't make those. Okay, somebody else defined those. For the primary analysis, we basically don't need to worry about that. It's from the original image and get the base call. So we know that which nucleotide is ACGT. And usually the software from the sequencer itself will help us to generate this. And for the bioinformatics part, we really start from secondary analysis, which include sequence alignment, sequence status. Okay, we want to calculate those. And consensus calling, we will talk about that. And create a QC file, quality control, how the data look like, and tag counting and sequence assembly. And all these things are considered as a secondary analysis. So you can, if you distinguish the secondary with the tertiary is there's not too much biology part kick in the secondary. So it's more like the data processing, basic data processing part is something that we will be dealing with today. And for the tertiary analysis, that will be very biological application specific. It can be DNA sequencing, including resequencing or de novo sequencing. It's a different workflow. And for the ChIP-seq, identify peaks or for the histone markers, whatever other things you can do. For the RNA sequencing, to calculate the gene expression, to calculate alternative splicing, to identify the variants on the transcripts, all those things. And for the epigenetics and for the DNA methylation, how do we further analyze the data? So most of the bioinformatics part, this part really needs to work together with the biologists and to make sense of the original data. And for the biologists, especially we have the students from School of Medicine and come here. And this part sounds like not important. And true, this part is something you do not have to run. Okay, and the bioinformatician will generate. However, it's really beneficial that you know what is the algorithm they use, what is the good thing and bad things about individual ones, so you can critically assess how well or how bad the bioinformaticians did, right? So don't just take us for granted. The data we gave to you, a lot of them are not really optimal, so you have to critically look at that. And in terms of where this analysis will be done, for the first part, it's usually done in the sequencing core, so we don't have to worry about that. For the second part, since it's not really biological application specific, and most of the pipelines is already ready, so bioinformatics should be able to take care of this part. And we are working with the bioinformatics core here and trying to pass some of the jobs to them as well. And for the data analysis part, this one is really a teamwork. So it needs the computer scientists, needs the biostatisticians, bioinformaticians together with biologists and to make sense of individual data. Okay? All right, the major steps for the secondary analysis, so I'm talking about the middle part. It's really not too much biological application related. And you got the raw data out of the sequencer, and this will be big, huge data. And you want to first look at the qualities, and you want to see the quality, how well the quality is, and what's the strategy you want to filter out the bad ones. And next step is alignment. So the so-called alignment is you got the reference genome. Okay, this is one of the sequence reads. You want to know this 50 base pair, which part of genome it comes from. And then you run through certain algorithms, then you figure out, oh, this one's coming from here, and this process is called alignment. There's some situations that one reads can align to multiple locations in the genome, and which happens very often because we know that human genomes are very repetitive. And the next step is the annotation part. So after you identify those biological signals, and then you want to know which part of the gene it is. Is that in the intron region of the gene or exon region? And those type of annotation is very important. And after that, and you send the data to the analysis team that we can start to make the biological stories. Okay, for the sequence quality. The quality is key, of course, right? So I got this from lab, and it's money, time, and quality. This is something that doesn't really come together sometimes. They are not agree with each other. And by sequencing a poor library will actually waste your money. That's for sure, right? And the sequencing reagent cost is huge, and cost of storing the raw data is pretty big, and CPU times requires is bad. And most importantly, the cost of analyzing this bad data are really cost you a lot of time and money. And there's some not very smart practice, but we see a lot of biologists do. And the first one is not checking the sample QC before the sequencing. So they get the samples and they send to the core facility, want them to do the sequencing. This is not smart. And one common mistake that people make is that they will skip the biological replicates. They thought this will save them money. Okay, when you run two samples, two conditions. Okay, I got MCF7, I treat with a certain drug, and I want to run RNA sequencing experiment. And so we know that if the experiment design is proper, and what we should is for each condition we get multiple samples, and then we do the sequencing, and then that offers us enough statistical power. But a lot of situations that people want to save money, and they just skip that replicates. They just sequence one sample from the MCF7, the other one from the drug-treated group. And that will causing the experiment much under the power. And but that can be solved by sequencing more duplicates in the later stage. But at this stage, you really need to be very careful. The experimental design needs to be balanced. So meaning that in the future, when you sequence, so you say, oh, I probably see some signals. I want to do more experiment on these samples or the similar conditions. And when you do the later runs, you want to make sure that you have both control and treated samples in the later runs as well. You do not want to say, the first run, I put all my control in the first run, and I put all my treatment in the second run. That's the common mistake people will make, right? And then you see the difference between two groups. You will never figure out whether that difference is because of the real biological difference or because some technician made some mistake or there's some variation in different runs. So you want to make sure the experimental design is really balanced. Okay, goes to the real thing of next generation sequencing. So what does this quality mean? So we often see that quality. And let me show you what this figure means first. So you can see this is a reference genome. Okay, this is a reference, and these are different sequencing runs, and they have already been aligned here. Okay, when we talk about the quality, in the future, I want you to keep in mind about that as well. When we talk about the quality, it's not one thing. Quality is not a word. Okay, it's here. So it doesn't mean anything. There's three different types of quality. So every time when we talk about the quality, we want to identify which quality we are talking about. The first quality is the so-called base quality. Okay, and for example, it got the first sequence tag here, so you can see the colors, and the colors really is encoded for the quality of that sequence. All right. So if I say this is an A here, what is the quality of that is A? All right. So that is called the base quality. Basically, tells you how accurate the sequencer sequenced to get this ACDT sequencer are. And the base quality will be for every nucleotide, and it will be reported by the sequencer. Okay? The second level quality is called the mapping quality, or in some conditions called alignment quality. So meaning that if I got this reads, the sequence reads, I want to align it back to the genome, and I say, okay, after my BLAST or other alignment algorithm, that I know that this read aligned here. So what is the probability that I made a mistake? Okay, so the mapping quality is very important as well. Okay, so if you say there's a two mismatch in this alignment, and how likely that will happen by random. So those kind of evaluations called mapping quality, and the mapping quality is one quality for every read, and this will be reported by the aligner. Does that make sense? The third quality we're talking about is the so-called consensus quality, which means after this alignment has been done, and I want to know whether this part is a variant or not. So is this a heterozygous variant or homozygous variant? And I also can calculate a p-value associated to that. And that is called consensus quality, which is a variant call quality. So we will have a consensus quality for every genomic locus that we have, and also this one is reported by the variant callers. All right, does that make sense? So keep in mind there's three levels of quality. The base quality reported by the sequencer, the mapping quality reported by the aligner, and the consensus quality reported by the variant callers. So we will go through all these different qualities. Okay? And the first one, before we go into any detail of any quality, And there's one thing called a Fred score, and it's used very widely in the next generation sequencing data process. Let me tell you what that is, okay? So the first Fred score was published in 1998 in these two papers, and they were both on genome research, and they were initially developed for the Human Genome Project. Remember that ancient Human Genome Project, right? And it's now widely used to characterize the quality of DNA sequence. All right, so some of the biologists probably see this type of figures all the time, especially we're in the genetics department. I have never seen this. I want to make it clear. I wasn't trained as a biologist. But for the people who does sequencing, and this is the figure they will stare at every day, okay? So you can see that there are different colors, and each color corresponding to A, C, G, or T. And at one location, if there's a green, that means this is A or things like that, right? So the point here is if you see there's a sharp peak of a color, and there's not other signals around there, and it's very clear that this is A or C, right? So your quality of that base call is very accurate. And for a location like this, you got multiple colors stacking together, and it's a fat peak, not really a narrow, nice, thin one. And the quality is not going to be very good. Okay, so the Fred score actually is to evaluate the base call quality for each individual location. And the definition is kind of simple, actually. So you can see that we are very familiar with the p-value or things like that, right? So if we know that p-value is 0.1, and that means our accuracy is 90%, right? If p-value is 0.01 and accuracy is 99%, so on and so forth. So the quality definition for the Fred score is minus 10 times log 10 p, okay? So it's just a convenient way to transfer these small numbers, more and more zeros in front, into this number that is nice and easy to look at, okay? So often that we are always use Q is 20 as a sum of the rule, and Q is equal to 20. That means the p-value is 0.01. That means that if I sequence 100 nucleotides, I had one make a mistake. By random, I will have one make a mistake. All right, I don't know which one, but I will have one make a mistake. So those are what the base call quality really means. Okay, so the point here for this slide is the quality score, and this is the so-called Fred scale, and which is a simple transformation for the p-value. Okay, so this will be used repeatedly. Okay, now we are talking about sequence alignment. Okay, the first sequence. So what is sequence alignment? It's a way of arranging the sequence of DNA, RNA, or protein to identify regions of similarity. And it helps to inferring functional structure of evolution relationship between the sequences. The goal here actually is to find out the best matching sequences. If you get one sequence, two sequence, and the best matching sequence, that's what your goal is. And there's two types of sequencing. One is the global alignment. The other one is the local alignment. So I don't want to give some strict definition on these things. Those doesn't make any sense anyways. So, but you can see these figures. This really tells you what global and local alignments means, right? For the global alignment, you want to, for these two sequences, you want them to align together. You want to account every single nucleotide. But for the local alignment, you just want to find the agreed regions between these two. Okay? And in terms of alignment theories, the goal of alignment is to analytically determine the best alignment between two sequences. Well, when we talk about this, is what is the best? This is a hard thing to define, right? And for the proteins, that we will have this type of very well-defined penalty matrix, the so-called penalty matrix. So you cannot see very clear, and I cannot see even from here. But this is 20 amino acid. This is another 20 amino acid. Okay, you can find out this in the lecture notes. And these are the numbers. So if it is perfectly aligned, there will be a positive number. And if it's a misalignment, there will be a negative number. And by the end of the day, that if there's a mismatch, so So because of the biochemistry features of different amino acids, so some of the mismatch has more penalty, others have less penalty, I think, so on and so forth. So you will need a scoring matrix or penalty scheme for this protein alignment. For the nucleotide alignment, it becomes much easier, and for the DNA or RNA, so you just basically give them scores, right? If there's a match, and you give a 1. If it's a mismatch, you give a 0. If there's a gap, you give a penalty. Okay? It becomes simple. So these penalty levels is the way that you can define it. Based on this scheme, I'm giving you this example. So you can see this is two nucleotide, two sequences, and clearly there's a gap here. And actually there's an 8 nucleotide match here. So you give this score is 8. And the minus, because there is a gap here, so you already gave a 3 penalty, minus 3. And every additional gap, you give a 0.1 extension penalty. So the alignment score between these two sequences will be 4.8. Okay, very simple stuff. All right, so you do need this kind of penalty scheme to define what is the best alignment, right? We were hoping that everything is perfectly aligned, but that's not going to happen. Yeah.
So the gap, 0.1, because you've got 3 gaps, you get 1 for 3.
Oh, for this one times 3 plus and this. So for if there's a gap, regardless, it's whether this is 1 base pair in the gap or a 3 base pair, you will get, as long as the gap created, so you got a 3 penalty. So every additional base, and you will get another 0.1 extension penalty. Well, these can be defined differently. Actually, different aligners can define this differently as well. This is just one example. Okay? All right. The goal of alignment is that there's 2 goals. One is a global alignment and the local alignment. The global alignment is alignment must account for all the characteristics of each sequence. Okay? Actually, it's not used in the next generation sequencing technology. And the algorithm to do that is called Needleman-Wunsch and algorithm. And for the local alignment, the goal is alignment accounts for only a continuous portion of each sequence, like this. And the algorithm itself is the famous Smith-Wortman algorithm, right? I wanted to just talk about Smith-Wortman algorithm, but later I figured out it's easier to talk about this first and then transition it to the Smith-Wortman. And the next couple of slides is from Dr. Shen Li from Center for Computational Biology and Bioinformatics, and he is trained as a computer scientist, and he's teaching the bioinformatics course in the School of Medicine. So this is a couple of slides about dynamic programming, how this global alignment works. So there's 3 major steps. The first one is the creation of a alignment path matrix, and the second one is the stepwise calculation for the prefix alignment scores and backtracking. You don't know what that means, all right? So let's get into it. So actually, it's quite simple. So you can see there's one sequence here, and there's another sequence here. Keep in mind, this is the protein sequence. For the nucleotide, there will be the same thing, okay? But I'm just using the protein sequence as an example, okay? For every gap, for this particular case, for every gap created, you will get a -8 penalty, okay? So you can see that initially, this one doesn't align to anything, So every additional gap, there's a -8 score penalty. And so is this one, all right? And what I'm trying to figure out here is fill in this penalty matrix, okay? So our goal is to calculate this number and based on the 3 numbers here. So if I calculate this number, I want to base on this, and the alignment performance. So that is the point. And the creation of alignment path matrix, so what we first say is you can see this is a P and there's an H here, right? And we got our penalty score from this blossom, the matrix, the smaller matrix I show you. For P versus H, the penalty is -2. So what I'm going to put in this number is this, okay? So for this number, it will be determined by these three numbers, okay? For this case, all right, I'm going to calculate from here, and then for this one, P versus H, this will be -2, right? 0 plus -2, so this is a -2 here. Or I could come from this path, which is -8. This is another gap here, which is -16. And or from here to here, this is -16. And the maximum value of all these three will be -2, okay? So what I'm going to put is I put -2 here, and I make a record that this -2 is derived from this particular location, okay? And then next, I'm going to worry about this value. So again, this value follow the same scheme. This value will be calculated based on these three numbers, okay? Now I have this P versus E, and the penalty is -1. Okay, so you got here is from this one, this will be the -8, because we calculated this -8. -1, this is -9 here, okay? And for this one, it's coming from here, -2, and the gap is -8, so this is -10, and -16, and the gap is another -8, so -24. And these three numbers, the largest is -9 here. And you just put -9 here. And also you want to record it that this number is coming from this path, okay? Now you calculate this one and follow the same strategy, but in this case, we have a tie here. Both are -10, so we'll still put -10 here, and I will put this record. So this -10 is derived from both these two, okay? So based on this simple strategy, and you will calculate this one, and I'm not going to go through it, and it comes from here. So based on this simple strategy, all the numbers in this matrix will be calculated, okay? And next step is you want to do the backtracking. So the so-called backtracking is you start from this location, so the score is is 1. So you want to see this 1, it comes from this part, okay? And then you see this is E, this is E, so you know that these two E's aligned together, okay? And then you come to this number -5, and this -5 comes from this part, and you know this is a gap here because it doesn't come directly from here. So you can see this A is aligned to a gap. And this -3 comes to here, 3 comes to here, follow this path, and this will be another alignment, which is E versus E. So you can put E versus E here. And then you step by step follow this path. Eventually, you will derive this potential global alignment. Let me finish the animation of this. Now you'll focus this, finish this alignment. So this is the so-called optimal alignment. All right. And so that is the Needleman-Wunsch algorithm. It's not difficult, but you have to sit down to think through it if you haven't heard about that. But the point here is what we really want for the next generation sequencing is so-called local alignment, right? So we got one sequence raise is about 50 base pair. We want to know which part of the genome come from. We are not going to align it against the entire genome from the very beginning to the end. We only want to find out the local regions that aligned. So the scoring system is exactly the same as the global alignment. The only difference is so the searching can start from anywhere. So if it initially it gets into the negative score categories, and it's always 0 here. And another thing is the searching can end at anywhere. Okay. So what happens is for the Smith-Wortman algorithm, the same matrix will happen. This is so remember, all these numbers were minus numbers, but if there's a lot of minus numbers, you just put a 0 here. and all the alignment only start from a non-zero score comes out, a positive score comes out. And this will be the Smith-Waterman matrix. And the backtracking part, it will start from the largest number within this matrix, and then you will further follow the same path here, and eventually you will derive this smaller region, the local region, where the alignment occurred. So you can see that based on this particular path, and this is the end of it because it reached to 0. And then now this is the optimal local alignment. Okay? So if you haven't, I know this cannot be done for just a few minutes, but if you want to think about that, and this is, you can go back to, and it's not that difficult to figure out. Okay, so the algorithm is there. There's a problem. So we know that Smith-Waterman or also the dynamic algorithm for the alignment, especially for the local alignment, we know it will guarantee give you the best optimal alignment. Okay, so that is has been proved the theory. But the problem to calculate this entire matrix is too costly. So if you think of this part is the sequence raised, this is the entire human genome. And if you really want to calculate this, only one read, how many numbers you need to calculate, right? It's just impossible to go through Smith-Waterman in this fashion. Especially we are talking about not one read, but trillions of reads. Okay? And therefore, fast alignment is become very, very important. So there's a fast alignment for short reads. Okay? For the short read aligners, there's some major challenges. The first major challenge is going through 1 trillion reads using dynamic programming is not practical. And one of the strategies to do this is to make a dictionary, or in computer science term, they call it index. Okay? So what happens is you have your old reference genome is already there, right? So from ACGT and to whatever is 3 billion letters. And then you want to find out, this is an example of a 4 nucleotide index. So I want to record all the locations of AAAA. Okay, there will be a lot of those, right? But you can make a dictionary of all the AAAs. Next time, when you got a sequence and you just grab the first 4 reads, okay? And then first the 4 nucleotides. Let's say it's AACA or AAAC. And then you go to your dictionary and to check, AAAC. And these are the genomic locations that have the AAAC there, right? But that is only taking care of the first 2 nucleotides. And you can still do Smith-Waterman algorithm starting from there. But that part, the searching will be in a restricted region, that not the entire human genome anymore. Okay? But the problem for this type of strategy is making a 50 nucleotide index is too large. We know that there's 50 letters, and each location potentially has 4 letters. And this scheme is 1.3 times 10 power 30. And also, most of algorithms, you need to load the entire index into the memory. It's just not practical. So making such an index is too huge. Okay? So it's not going to work. There are a couple of there's a couple of strategies that can help us for the short read aligners. But before we get into that, we need to know what we are dealing with. And the features of these reads is short and massive amount. And the cost here we need to consider, which is the speed, how fast you need to do the alignment, and also the resources that are required. The resource here meaning the memory, right? We know that for most of the laptop, you have 4 gig base memory, maybe 8. And for desktop, you got a little bit more. But for the servers, and the largest server in my lab is only 48 gig memory, right? So we know that university have a half a terabyte of memory. That's supercomputer. But memory is an issue. So you need to consider how much memory is required for each alignment algorithm. And there's another one is for the alignment quality, meaning that there's a lot of fast aligners they do not allow gaps. Okay, so that's one of the ways they make their alignment faster. And if the gaps are something that you're concerned about, and those are the algorithms you cannot use. And we will go through those as well. And information considered, some of the algorithms just strictly get the ACGT numbers from the read and do the alignment, and other algorithms rather you also use the base causality. So basically, if you got a 50 base pair nucleotide, you align to the genome, there's 3 nucleotides mismatched, but 1 or 2 of them are the base quality is not very good, and you probably want to discount the contribution of those 2 nucleotides. So some of the alignment algorithms do use the base quality information, others do not. And accuracy as well, and we have a couple of slides about that. So there's a couple so I'm not trying to teach you any specific short race aligner. They are all very well done, okay? But I want to, in terms of theory-wise, I want to summarize what type of strategy that people start to take in terms of solving that the problem that we had, right? The problem is that we have massive amount and dynamic programming is too slow. And the first common strategy is a hash table-based strategy. This is the so-called seed and extension paradigm. So you can imagine here, this is the sequence read, 50 base pair, and this is the reference genome, okay? You cannot really index the entire thing. 50 base pair is too big. Rather, you only index the very beginning part of it. It's called a seed, okay? If your seed is long enough, and you can create enough specificity on the genome. So what you do is you first index the seed, and then you align the seed back to the reference genome, and you further start from there, because now you don't deal with the whole entire human genome, but rather very small parts that's very close to the seed part. And then you further do the extension and to calculate how well these 2 sequences matches. So you can see that I make it a little faint, saying this 2 doesn't really align well, and this one really aligns strongly, and that will help you to speed up the alignment process. Okay, very straightforward way of thinking. Okay, and for the suffix, and there the second strategy is called the so-called suffix tree and prefix tree, and suffix array or Boros-Wheeler transformation. We'll get into that, and so it's a lot of fun to go through that. And the third one is called merge sorting, and this is not really commonly used, so I'm not going to talk about that. So these 2 are the major things, okay? And most of the current published 24 also aligners, they are either hash table based or suffix tree based, okay? So you can really pick the ones that you want to use. For the hash table based and the algorithm, so let's have a few slides about that. Then the first one is the feature is very straightforward. Okay, so you got so we mentioned that we got this is a dictionary. You just make this dictionary. It will sit into your memory, and anytime you see a nucleotide, you want to go that place and to find and then to do the extension, right? And the size is a full power k records for k more hash table. So if it's you got a 4 nucleotides, that will be 4 power 4. If you want to build a 15 nucleotides hash table, and that is about the size that most people are using, or 14 or 13, and that will be 4 power 13. And that is still workable. So if you make the calculation, they can, that size of the seed can really sit in your memory. That's no problem. Okay, so the seed extension paradigm. So you can imagine that this is the query sequence and the seed, and we make a pretty good dictionary, and then go back once I start from the seed, and I know there's maybe half a dozen locations in the genome has the seed, and I further do the extension to calculate how well it match. Okay, and there's a couple of algorithms using this hash table-based algorithm. Some of them are very widely used, and including the ELAND, which is indexing the read. There's 2 sub-strategies. The first one is indexing the read. The other one is indexing the genome. and to index the read, and the most famous one will be the ELAND algorithm. And I'm not going to talk about that because we are not allowed to talk about that. Not really, but we don't know the details. It's commercial software, and they never published their algorithm. And that is the one that Illumina was using very initially, okay? And MAQ is a hash table base, and Mosaic and they both are hashing the reads. And there's also some others hashing the reference genome, including BFAST, Mosaic, and SOAP, okay? And BFAST is really the one that we are using in my group right now, okay? All right. But you may think, if you think deeper about this, okay, if perfect match is straightforward, right? So if I want to find, if I got a nucleotide, 50 base pair, I want to find it in the genome, where it comes from. If everything is a perfect match, it's pretty simple. But it's not useful to identify genetic variants for the hash table-based study, right? Keep in mind, if there's a SNP happens in the seed location, okay, the initial seeding will be missed. You will never find that nucleotide anymore, that alignment anymore. That causes a lot of trouble because one of the points that we are doing sequencing is to identify genetic variants, right? So the algorithm needs to take care of it, and here's how. The solution is to use the multiple the reason I talk about this is while I was reading all these things, and those are the questions that come into my mind, so I have to read deeper to find out how they solve it, okay? So I want you to just follow this strategy. The solution here is using multiple indexes that allow mismatch. One example is here. So if I have this 16 nucleotide read, I want to do the alignment. If I do not allow any mismatch, I can make a 16 base pair hash table, right? Just straightforward, done. However, if I allow one base pair mismatch, and this will be the strategy I'm taking, okay? So I'm going to only hash the last 8 or the first 8, okay? And there's the zeros here. The zeros are the nucleotide positions that I'm not going to consider. So what happens is for any string with one nucleotide mismatch can be covered by at least one of these two indexes. Okay? If the mismatch happens here, and this index will still cover that. This is really for the sensitivity issue. If you do not allow mismatch, your sensitivity goes dramatically down. So by this way, using these two indexes schemes so that you can really take care of or allow one nucleotide mismatch for anything happens. Does that make sense? Okay? So any string with one nucleotide mismatch can be covered by at least one of these indexes. So this matrix is called a mask. Okay? You have to know about it. And there's a specific ways to design this mask, which is very interesting. Okay, we got a mask, but there's more than one way to build the mask. Let's go through this. So you still get this 16 nucleotide read. Okay, so this is our way to build the mask so that you can allow one base pair mismatch. Okay. And there's several different statistics in there. The first one is M, which is the seed length. The seed length is the 16 base pair. Okay, K is 1. K is the number of mismatch allowed. And the W is 8. W is within each seed, how many ones we have. So meaning that how many nucleotides I'm counted to build this index. For this one, W is 8. And N, which is the number of index, is 2. So we got 2 indexes that can cover everything. But this is another way to build the index, right? So this is also a valid scheme. So you get so the point of this index is really if I have one nucleotide mismatch, and I can find it from any one of these indexes. And this is another one, and which is probably a better one to a certain extent. which you can see that the W is equal to 14, meaning for each individual index, there's a 14 nucleotide has been counted. However, you need 8 such indexes to cover everything. Okay? So the point here is if there's one nucleotide mismatch from the original seed index, this original read, and you will be able to find it from at least one of these indexes. Okay? All right. But we are having a problem. So we have this 16 nucleotide, and there's multiple ways to build these indexes. But we have a problem here. The first problem is the seed weight, W, should not be too small. So you remember we got a 16 base pair nucleotide, and when we break it into 2, our weight becomes 8. All right? The problem of 8 is too small to avoid many false positives that will allow the mapping process. Okay? My initial goal is to build a 16 nucleotide index. All right? So maybe there's 3 places in the genome has it. However, if I reduce it to 8, and there probably is 10,000 locations in the genome has it, and there will be a lot of false positives, at least for the seeding part. Of course, you can get rid of those after the further alignment, the extension part, but that will take a lot of time to figure those out. So that will slow down the process. But the higher the seed weight is, the more seed would be needed to achieve full sensitivity. So you can see that this one, that the weight is 8, you need 2. The weight is 14, you need 8 such kind of indexes. And if you have things like this, you run into another problem that you have too many indexes needs to be loaded into the memory, and that eventually will also slow down your mapping process. Okay? So how to design those masks really, it was a topic a couple of years ago. Yeah. So you're talking about shifted seed in the read, is that right? Okay. So the question is, if you got a 50 base pair arrays, you design a 16 base pair seed, and so we're talking about the seed and extension. You first use the first 16 nucleotide, and then you do the extension. And Jake's point is, why don't we do the shifting and to account for this? And that will be also very complicated. So most of alignment algorithm that requires 2 nucleotide mismatch, okay, allow 2 nucleotide mismatch. So the notion for that is there's 1 SNP, and also allow 1 nucleotide for sequencing error. Okay, so once you got 2 nucleotide mismatch, and that 2 nucleotides can happen anywhere, and how do you do the shifting? Will become an issue. You may end up spending more time to figure that out. Okay? But let me go through this so you will know there's an optimal design for this, which is really, really nice. And the question here is, does an optimal design exist? Actually, it does. Okay? And so you can read that later, but let me give you the point here. There is a beautiful work from Chinese Anatomy of Science and from 2008. So what they do is they publish optimal mask design algorithm. So what happened, and they also build this into their aligner they call it Zoom, zillions of oligos mapped. Okay, and it's not really supported anymore, but it was a very academic way to solve this problem. So what happens is they give you the best design, the best optimal solution. So you can see this table. You cannot see from very well from the back, but you can see this is the read length that you are targeting. For example, this is 33 or to 36 to 25, and this is the mismatch allowed. Is 2 mismatch if you allow 2 mismatches, and this will be the number of indexes needed for a specific weight. So let's say if for let me go through it. For 33 nucleotide read, okay, if your weight is 14, and you can look at this table that you know that you need 4 index, you can find out a way to design 4 index to cover everything that have 2 base pair mismatch. Okay, here's the proof. Okay, so the seed length is 33, and the weight is 13. Okay, mismatch allowed is 2, and this number is equal to 4. So you need this 4 index to cover all this reads, and at the same time allow 2 mismatches. So actually, I stared at this for about 10 minutes and trying to prove they're wrong. Okay, so basically I was trying to say, here these 2 nucleotides are mismatched, and it will always be taken care of by one of the seed indexes in here. All right, so we don't have to do this now, but if you find any 2 mismatch were not taken care of by any one of it, let me know. We have to get their paper withdrawn. All right. But I think this is a really very interesting academic way to solve this problem, and this was published in 2008. And actually, many later aligners using this type of strategy to design their matrix, mask matrix. Okay? So I guess this is a fun practice, right? So you got the seed, we know the seed extension, and we know the problem. If you only focus on the seed, you're running into trouble because you are not allowing nucleotide mismatch into the seed. So using this type of mask design can help us to solve it. So if you, for the students in the room, by the way, that we will have homeworks, okay? And when we do the homeworks, and I'm going to ask you to do to install the aligners, so you will realize, I thought there's one index, only one index. Actually, there are some aligners that are 10 indexes, and this is what happens, right? This is the way that they design the mask and matrix, and that can help us to take care of mismatches. And here, it's a simple thing. I'm only talking about the mismatches. Some of aligners, they were trying to tolerate gaps in this and to design some other novel matrices, and it's even more complicated. But the point is here. Any questions so far? Are we still following? All right, good, hopefully. All right, and the hash table strategy is good. So you got a seed extension, okay? And the problem of a hash table-based strategy is alignment to multiple identical copies of a substring in the reference must be performed for each copy. So what I mean here is, we know that human genome are very repetitive. So there is a lot of regions that happens many times, okay? And for this type of method is once you got a seed, and then you map it, you use the seed to identify the potential locations further do the extension. And if there's really a repetitive region, for every single location, you really need to go through this Smith-Waterman algorithm, so which can be very slow. Okay, so there are some solutions for that, which is use a suffix or prefix tree that can handle this. How many of you have taken the algorithm class in computer science? So you heard about this suffix tree, prefix tree, right? I've never heard about that before. And I'm not going to tell you that how to do it, but let me tell you what I understand about it, and I think that will be enough for the biologists. All right. So what happens here is this is becomes your reference genome. Okay? It's weird, I know. So what happens is you got a reference genome, which is 3 billion letters, and that you can really generate a huge tree, and that represents this reference genome. I'm not going to tell you how to generate that tree, and it's complicated, but it's doable. But the point of this is searching become very fast, very fast. So let's go through this, okay? So what I talk about is, let's say our reference genome is only 4 base pair and 6 base pair. And this 6 base pair is extended to this huge, humongous tree, okay? However, let's do the searching. So if I want to search AGG, whether AGG is in here, so what happens is you always start from the node, the root, and then you search from the back. I'm searching AGG. So you will search G, G, A. All right? So you can always search from the back. So everything that from the root, you will have a path to there, okay? And the computer scientist told me that this gives you a very fast searching scheme, which is ON, which N is the size of your read, not the query sequence, which is a 50 base pair, not the length of the entire human genome, which can be very, very huge. So the searching part is very, very fast. Okay, you probably still don't know how this works, and I'm not going to explain that because I don't know either. But that's okay. So it's not going to be used anyways. And there's a bigger problem of this tree is I have to design a 6 nucleotide, and this tree will become this big. It's impractical to make the index for the entire human genome into such a tree. Okay? But this, the point of this suffix tree and prefix tree is the searching, the query part. Once the index is established, which index is this tree, and the searching part, query, become really, really, really fast. Okay? Well, for the Smith-Wortman, as you can imagine, for every sequence alignment, what I have to do is to generate this big matrix and do the backtracking, and that's not going to happen, especially one of the read sequence here is the reference genome, which is 3 billion letters. But for the suffix to prefix tree, and to do this searching is extremely fast. Okay. I'm going to take a 10 minutes break, and coming back, we'll talk about the Boros-Wheeler transformation. And this is something it's very, very popular right now in alignment algorithm. And the most important ones, the Bowtie and the BWA, they were all based on this. And for the biologists in the room, you probably don't even know these type of things. But however, I still need to talk about that because if you're an informatics student, you want to look for jobs. And I'm afraid this is one of the questions that they want to ask you for the job interview. So after we come back, we'll go through this Boros-Wheeler transformation. Okay? 10 minutes break.